Assalamu alaikum. Our next topic is visible absorption spectrophotometry. Ultraviolet and visible absorption spectroscopy is the measurement of the attenuation of a beam light after it passes through a sample or after reflection from a sample surface. This property is influenced by the sample composition, potentially providing information on what is in the sample and at what concentration. Since this spectroscopy technique relies on the use of light, let's first consider the properties of light. Whilst there are many variations on the UV vis spectrophotometer to gain a better understanding of how an UV vis spectrophotometer works, let us consider the main components. So, first component is the light. So, light as a light based technique, a steady source able to emit light across a wide range of wavelength is essential. A single xenon lamp is commonly used as a light intensity light source for the both UV and visible ranges. So, wavelength selection. In this next step, certain wavelengths of light suited to the sample type and light for detection must be selected for sample examination from the board wavelengths emitted by the light source. Available method for this include monochromator. A monochromator so monochromator separates light into a narrow band of wavelengths. It is most often based on diffraction grating that can be rotated to choose incoming and refracted angles to select the desired wavelength of light. Absorption filter. Absorption filters are commonly made of colored glass or plastic designed to absorb particular wavelength of light. Interference filter, also material where interference occur between the thin layers of materials. These called dichroic filters. These commonly used filters are made of many layers of dielectric. A filter can be used to eliminate undesirable wavelength by destructive interference, thus acting as a wavelength selector. Cut-off filter. Cut-off filters allow light either below or above a certain wavelength to pass through. Band pass filter. Band pass filter allow a range of wavelengths to pass through that can be implemented by combining short pass and long pass filter together. Detector. Detectors are devices that indicate the existence of some physical phenomena. Some example of simple detector are transducers, photodetectors, photographic films, mercury level in thermometers, and human eye. Transducers. It is a special type of detector that converts signals such as light intensity, pH, mass, and temperature into electrical signal. Photodetector. Photodetector used photo tubes, photo multipliers, tube silicon diodes, photovoltaic cells, phototubes. So here's the application of this process. Application UV spectrophotometer may be used as a detector for HPLC. The presence of an analyte gives a response assumed to be proportional to the concentration. For accurate result, the instrument's response to the analyte in the unknown should be compared with the response to standard. This is very similar to the use of calibration curves. The response for a particular concentration is unknown as the response factor.